Hi, I'm Perry O'Tierney-Ginn, Assistant Professor of Reproductive Biology at Case Western Reserve University in Cleveland, Ohio. And I'm here in my lab at Metro Health Medical Center to tell you about our newest data published in the August issue of Endocrinology. In this paper, we studied the placentas of nearly 100 healthy, lean, and obese women and found that their placentas metabolized lipids very differently. Lipid metabolism in the placenta affects several key processes for placental function: steroid production, ATP generation, inflammation, and potentially lipid transport to the fetus, which is critical for proper brain and cardiovascular development. Maternal obesity, affecting 65% of deliveries at our hospital, impairs or aggravates many of these processes. So we asked how or if lipid metabolism is altered in placentas of obese women. I'm going to highlight two of our findings. First, placentas of obese women have a greater lipid content. These extra lipids are stored in lipid droplets, visualized here using immunofluorescence staining for perilipin-2, a component of lipid droplet membranes. The lipid droplets can be seen in green, red is cytokeratin 7, a marker of trophoblasts, and the nuclei are in blue. We found that the lipid droplets are localized mainly to trophoblast cells of the placenta, the sites of metabolism and transport. We're the first to show that this extra fat in placentas of obese women may be due to the upregulation of the fatty acid esterification pathway shown here. In a smaller cohort of women, we measured the rates of esterification and storage of a radioactively labeled fatty acid palmitate in vitro. Trophoblast cells isolated from placentas of obese women esterified the fatty acid tracer 50% faster than trophoblasts from lean women. We think that this could be at the root of the high inflammation and oxidative stress found in placentas of obese women, a process called lipotoxicity. Besides being stored, fatty acids are also metabolized for energy in the placenta, and we wanted to understand how obesity altered fatty acid oxidation. 90% of fatty acid oxidation occurs in mitochondria. We found that there were fewer mitochondria in placentas of obese women. However, other organelles, such as peroxisomes, can partially oxidize fatty acids and may help to compensate for impairments in mitochondrial function or number. But how do you measure their separate contributions? To do this, we use the CPT1 antagonist atomoxir, which specifically blocks mitochondrial fatty acid uptake, thus exposing the contribution of non-mitochondrial sources of oxidation, such as peroxisomes. Atomoxir blocked cellular fatty acid oxidation by 90% in lean women, but only 80% in obese, showing that non-mitochondrial organelles were responsible for 10% more fatty acid oxidation in placentas of obese women. Thus, overall fatty acid oxidation was maintained in placentas of obese women despite reduced mitochondrial capacity. Isn't the adaptive capacity of the placenta incredible? What do all these changes mean for the fetus? We aren't sure, but we have some clues. We found that higher placental lipid content is correlated with lower neonatal adiposity among obese women, suggesting that increased placental lipid storage partially protects the fetus from excess maternal lipids. However, this may come with the cost of lipotoxicity to the placenta. We'll pursue this train of thought in future studies. Thank you for listening. We hope you read our paper to get more details. And for more information about our work, please visit our lab website at placentascience.com. You can also follow me on Twitter @placentasci.